Jorge is a very bright, up and coming star in our field. He spent a substantial amount of time doing his PhD with Susana Martinez Conde back when they were in Arizona. Then Jorge joined David Z and Amir Karadman, their group, to work as a postdoctoral fellow, where he was awarded a very prestigious NIH R00 K99, which is considered a very prestigious grant to launch his independent career. Now he is in University of California, Berkeley, running his own lab. His forte is microsaccade, his master microsaccade. So with that, let's start. Jorge is not around. I'll text him if he's available for thing. Otherwise, we'll move on. Jorge's, we can start his lecture.
Hello, I'm Jorge Otero-Millan, an assistant professor at the School of Optometry in the University of California at Berkeley, and I want to thank the organizers for inviting me to talk to you today. And I'll be talking about microsaccades and psychotic intrusions during both normal and pathological visual fixation. So I'll start by giving some background on just why our eyes move and why do they do it the way they do. Then I'll go over microsaccades, saccades, and psychotic intrusions, what's different, what's the same about them. Finally, I'll give some examples of how microsaccades can be used as biomarkers for visual problems, for motor problems, and for cognitive problems. And in each case, I'll also show some basic science results that can be related. So first, why do we move our eyes the way we do? I like to think about it as a method to compensate for the limitations of the retina. If the retina could see the entire world with high resolution, no matter the motion or blur, we wouldn't need to move our eyes. But that retina probably would not fit in the space of our eye. So first we have spatial limitations. As I said, only a little portion of the retina can see the visual scene with high resolution. So the eyes need to move to relocate this high resolution area around the object. And the main eye movement here is saccades. But then there are also temporal limitations. If the eye is moving fast, the image will be blurred in the retina and perception will be degraded. So we need eye movements to continually try to stabilize the image when we are moving around in the world. We have the vestibular color reflex, the optokinetic reflex, or the smooth pursuit if the object is the one moving and not us. So, spatial limitations, as we said, there is a phobia, this little area with high density of cones that will let us see and recognize high detail, but in the periphery we need much more. A bigger size of letter to compensate for the low resolution. And an example of the temporal limitations comes from fading. So on one end, if things are too still in the retina, they will disappear. So if you fixate here at the red dot for a while precisely and you keep your eyes on it, at some point the blue disk will disappear. So if we don't move our eyes enough, things disappear, perception degrades. And on the other hand, if things move too much, things will be blurry and they're hard to perceive as well. And finally, there's another temporal limitation that is masking. And I'm not gonna show a demo, I don't think it'll work through video, but you can imagine that you flash a stimulus first, and a stimulus that is visible enough by itself, but if shortly after you show another stimulus, this stimulus can become invisible. So if things are flashing too fast, or if a stimulus is changing too fast, some stimulus can mask others. So all these limitations determine how we move our eyes. First, the eyes need to move to compensate for the reduced area of high resolution in the retina. But they cannot move continuously because they would cause continuous blur. They must move in jumps. So you want to have quick jumps and then periods of relative stability. And those jumps should be separated for enough time so masking doesn't become a problem. And there is enough time for visual processing to happen. And finally, they must not remain still for too long so things don't fade and disappear. Okay, so now I'm going to go some of the work I did with Susana Martinez Conde. So first I'm going to talk about two behaviors, attentive fixation and visual exploration, and compare the eye movements that happen during them. So when we try to fixate in a dot, our eyes are never perfectly still. We have microsaccades, drift, and tremor. Here is an example of how those look. So we can see here those quick jumps, those will be the microsaccades. This is not a real recording, this is just for demonstration. And in between microsaccades, we have this slow drift with superimposed tiny little tremor. Okay, so this is a very nice demo to see your own fixational movements, to see your own microsaccades. So if you fixate at the black dot for a little while without moving your eyes at all, you just keep fixating on it, that will burn an after image of that grid. And now, if you move your eyes after you've been fixating in the black dot for a bit to the white dot, then you'll see an after image of a grid that will keep moving. So that movement corresponds with the movement of your eyes during fixation. We're usually unaware of it, but these after images can make it visible to us. So, on the other hand, on visual exploration, that's the behavior we do when we are looking around. And again, as I mentioned before, we are placing our fovea in different parts of the image. So we only see a little bit of high resolution at a time, but our visual system creates the illusion that we can see everything with high resolution by connecting these little snapshots that we see with each eye movement. So this is a recording actually of eye movements while somebody explores this image, and we can see the quick saccades followed by short periods of fixation. So I'm going to show the results of some studies that were trying to ask these questions. Are microsaccades generated by the same neural mechanism as saccades? And if so, can we say that fixation and exploration are actually equivalent behaviors? And this will become more clear as I go on. So we can think that microsaccades are random and serve no useful purpose on one end. We could think that each microsaccade is generated with a given purpose, but probably we're more somewhere in between, where microsaccades are part of the overall optimal sampling strategy of our visual system. So in this experiment, we had subjects doing visual exploration of different types of images, just a gray scene, a natural image, picture puzzle where they had to find difference, and a Where's Waldo image where they had to find Waldo. And we also had conditions with the same images where they had to fixate in a cross. And then we recorded all the movements in this experiment, and here we are plotting all the saccades during the visual exploration conditions. So each dot corresponds with one saccade, and we have thousands here, and the x-axis represents the amplitude, the magnitude of that movement, how big it was, and the vertical axis represents the velocity, how fast the eye moved. And this is a known relationship typically called the main sequence. Now in red here we're showing the microsaccades that occur during fixation. And we can see how they follow the same main sequence. Then we can also look at the timing. So we can plot the distribution of intersaccadic intervals during visual exploration and during attempted fixation. And we see that on this side of the distribution, they overlap, meaning that there seems to be a similar refractory period, or a period after a saccade or a microsaccade where you don't make any other eye movement, that it's governed by the same mechanism for both saccades during exploration and microsaccades during fixation. On the other end, there is a big difference because microsaccades are much less frequent. So there are bigger intervals in between microsaccades than in between saccades during exploration. But with this experiment, we see that both microsaccades and saccades follow the same peak velocity magnitude relationship or main sequence, and they have a similar refractory period. So this makes us think that the same circuit triggers both saccades and microsaccades. And this was confirmed later by some studies that recorded in different areas of the brain, including the superior colliculus, or previously in the reticular formation, where they show equivalent activation for saccades and microsaccades. So we went a little further and trying to dig deeper into this similarity between exploration and fixation. So we can think of it as exploration as a sequence of saccades interleaved with drift, but fixation is also a sequence of microsaccades interleaved with drift. And we think that saccades and microsaccades share a common generator, so could it be just behavior are the same, and fixation is just exploration of the dot. So what's different? As we said, the interval between saccades, or the rate, how frequently we make saccades. We produce about 3 saccades per second during exploration, but only 1 microsaccade per second during attempted fixation. So we did an experiment with the hypothesis that saccadic rates would decrease the side of the field of exploration decreases, with a continuum with classical exploration on one end and fixation on the other end. So in this experiment, we have natural zine of different sizes, and at the end, a tiny fixation dot. And we measure the saccade rate. So for the big image, we have the higher rate, and for the fixation spot, we have the low rate. And as we decrease the size of the image, the saccade rate decreases until it gets to similar one of the one during the fixation. So now the question here, there could still be an alternative hypothesis. So we could think of a bimodal distribution where saccades and microsaccades are generated independently. So then as the image decreases, it's just the proportion of big saccades decreases, but the microsaccades are always the same. And that would be consistent with our data. But we think it's more likely that there is just a continuous shift of a single distribution of saccades. So we check for that, of course, and that's what we found. We find this unimodal distribution of saccade amplitudes, and the distribution is just adapting to the size of the image. We don't see a bimodal distribution between saccades and microsaccades. So we think of it as scanning at different scales. No matter what you're looking at, we are just doing the same behavior, scanning with a sequence of saccades and drifts. When we observe small things, we scan them with less frequent and smaller saccades. We look at big things, we produce larger and more frequent saccades. Okay, in this next part, I'm going to talk about psychotic intrusions, how visual fixation is constantly interrupted by fixation line movements. But in the clinical domain, we would usually refer to them as psychotic intrusions when we see them in patients. And square wave jerks are the most common type of psychotic intrusion, and they are characterized by one small saccade that moves the eye away from the fixation target, followed by a corrective saccade that moves it back shortly after. And they are prevalent in many neurological disorders like PSP, but they are also found in healthy subjects. And this was work in collaboration with John Lee and also Susanna Martinez-Conte. So here's an example of a square wave jerk. Give me a second. So this is an example of a square wave jerk in a patient, and we can see how the eye moves back and forth, but there is always a little fixation in between. And this is an example of a recording of a square wave jerk, and this is what gives the name, this sort of a square shape. So we have one saccade that moves the eye away, and another saccade that brings it back. And this is the eye position over time, and this is just the X and Y display of those movements, which in this case, as usual, are mostly horizontal. So microsaccade and square wave jerks were considered different oculomotor phenomena, but usually because square wave jerks were studied in patients, whereas microsaccades were studied in healthy. And also because microsaccades tend to be smaller than square wave jerks. But it was suggested that they're probably just part of a continuum, and that's what we wanted to study. So we had to develop a method to quantify and identify and quantify square root years and microsaccades, and we tried to test the hypothesis that they fell in a continuum. So we had patients with PSP and a set of control subjects. And we identified all saccades up to 5 degrees, and we had a criteria to qualify as square root years. So first we see what we expected, and we knew in this population that the main sequence, the slope of the main sequence is smaller. So something is wrong in this patient that makes all saccades be slower. But what appeared to show us this continuum is that if we measure how likely a microsaccade is to be part of a square root year, we see a relationship with magnitude. So if we classify microsaccades, some as being part of a square root year and some as not being part of a square root year, the likelihood of being part of a square root year grows with saccade magnitude. So the larger it is, the more likely to be paired or coupled with another saccade to bring the eye back. And this is the same for controls and patients. And here's just an example trace where we have a larger microsaccade coupled with a return forming a square root year, and other small ones that are just by themselves. And we can show this also again by looking at intersaccadic intervals, so we can measure the intervals inside the square root years and the intervals outside of the square root years. And we see that this time inside the square root years is always shorter than outside, but that time was more or less equivalent between PSP and controls. So it seems like it's just more likely that microsaccades are larger in these patients, and that couples them with the return saccade, but that return happens at the same time as it would happen in healthy subjects, even if less often. We tried other populations of patients, and we found the same result, equivalent in intrasaccadic intervals, and a relationship between magnitude and likelihood of being in a square root year. So again, from this part, as microsaccades become larger, they're more likely to be followed by a corrective saccade, forming a square root year. And it seems that microsaccades and saccadic intrusions are just the same phenomenon, and that the square root years are generated by a common coupling mechanism in PSP patients, other neurological disorders, and healthy observers. Okay, so now I'll go quickly through a few examples of microsaccades as biomarkers. First, for example, in visual problems like amblyopia, the work of Fatima Gassia and Asif Sheikh and others has shown that if you measure microsaccades at different degrees of severity of amblyopia, the amplitude of these microsaccades changes, and they become larger as the amblyopia gets more severe. And we measure these eye movements in healthy controls also by testing the effect of a virtual scotoma. So we had subjects exploring an image, but there was a little portion of the image that was blocked. And as they moved around, that blocked area, that virtual scotoma, followed them. And what we see is that when you have the scotoma, when you have nothing to fixate on at the center of your fovea, you make less small microsaccades of the size of what you would expect as saccades. Then, as we showed before, microsaccades seem to be affected in some neurological disorders with more frequent square root jerks. So we can use microsaccades to distinguish, for example, potentially between control patients, Parkinson patients, and PSP patients. So the saccade rate is elevated in both disease populations, but the saccade magnitude is only increased in PSP, not in Parkinson's. And the main sequence slope, it's also only affected in PSP, not in Parkinson's or controls. Then, in a study done with my colleague Leandro Di Stasi, we looked at healthy subjects and when they were doing a task that was exhausting and long. So over time, subjects get fatigued, and we can use microsecond, in this case velocity, as an indicator of fatigue. So over time, like these were different hours doing this task that simulated like a air traffic controller, the slope of the main sequence would decrease over time. So saccades would be getting slower and slower. Finally, microsaccades can also be used to study our cognition. For example, in ADHD, the work from this group showed that in healthy subjects, if you show a stimulus and you measure microsaccades aligned to the stimulus, you would see that there is this periodic suppression of microsaccades. So if here you have a row with each trial and each dot is a microsaccade, you can see that once you complete the picture of all the trials, microsaccades are periodically suppressed. On the other hand, in ADHD, you don't see such suppression. So the mechanism that suppresses microsaccades when there is the expectation of an stimulus or when some stimulus onset happens is not present in ADHD. In the lab, we studied the effect of cognitive load in microsaccades. So we compared what happens to eye movements with just fixating and doing a mental arithmetic task. A very easy task where you just count by 2, and a difficult task where you count backwards starting from a 4-digit number in steps of 17. And what we see is that actually they're very effective. So in the hard task, here in red, and here we see that microsaccades are less frequent and larger compared to the green one, which is the easy task. Now the gray one is when they were not doing any task. So somehow, I think of this as you're paying less attention to the fixation spot, you're making less effort to explore the fixation spot, so you make less frequent microsaccades. So to summarize, microsaccades and saccades are indistinguishable and probably generated by the same mechanism. Attempted fixation is equivalent to exploration, just at a smaller scale. And the squirrel jerks are a particular case of microsaccade pairs, just affected in some patients. And microsaccades can be used as useful biomarkers to track progression of visual, motor, and cognitive disorders. And thank you so much, and I'm happy to answer any question you may have.
You had to open the microphone.
Yeah, got it. That helps. There was a question from from someone in India that there was a condition where there is a spinocerebellar ataxia with lots of microsaccades and wanted to know the name. So that is CASI, SCASI. That's a spinocerebellar ataxia with psychotic intrusions. Jorge.
Yes, Jorge, you say in amblyopia, the microsaccades are of much larger amplitude. It doesn't matter if the eye is iso or exotropic, is it?
So, yes, so it's basically strabismic or non-strabismic amblyopia. In both cases, there was an increase in this microsaccade amplitude, and that correlated with the degree severity of amblyopia. So it was more of a function of visual deprivation, not the strabismus. There was another paper that we did where we had strabismus, strabismic patients, strabismic amblyopes and their microsaccades. And the issue there was the trajectories were off. And also other interesting thing about amblyopia was that there was also a impairment of microsaccades when fellow eye was viewing, not just the amblyopic eye was viewing, because someone can argue that if you are viewing with the amblyopic eye, so you are visually depriving someone, and then it's because of the visual deprivation you are getting it, but it's not really just the visual deprivation because fellow eye was also abnormal.